So good afternoon, everybody. We are here today with Professor Aldrin Gomes. He's a professor of neurobiology, physiology, and behavior, and also a professor of physiology and membrane biology here at UC Davis. And he is originally from Trinidad and Tobago. He went to undergraduate school there, and he got a BS in biochemistry and zoology from the University of the West Indies in Trinidad. Then he performed his PhD also there. It's a PhD in biochemistry, also in the University of West Indies. And then he performed his postdoctoral fellowship in the University of Miami School of Medicine. And Dr. Aldrich studied drugs that are used to treat cardiovascular diseases, and specifically he studied the effect of these drugs in the cascades, intracellular cascades, and also mitochondrial function in cells. So today he's going to talk to us specifically about ibuprofen, and his talk is entitled The Dual Nature of Ibuprofen, Benefits and Precautions. So thank you very much, Aldrin, for being here with us today, and please go ahead. Thank you.
Thank you. So as Veronica said, I am from Trinidad and Tobago, also known as the Caribbean, and I've been at UC Davis now 15 years as of this month. So it's quite a nice time here. And today I'm going to be talking about ibuprofen and the dual nature of ibuprofen. Now, ibuprofen is one of at least 20 NSAIDs in America. And around the world, there's even more than 20 NSAIDs. Even though you probably only know of a few, there are a lot that are available, especially by prescription. And it's part of something called a nonsteroidal anti-inflammatory drug, meaning that it's not steroidal, but it also has anti-inflammatory features. And this is what distinguishes it, makes it different from something like Tylenol, which is acetaminophen. That is not anti-inflammatory, even though it's pain-reducing and fever-reducing. So there are two main types of NSAIDs. These are non-selective, like ibuprofen, naproxen, and then they're selective, like celecoxib. Now, what does ibuprofen look like? You can see it there on the left. Ibuprofen, we commonly buy it in America as Advil, but it's known by over 200 different names around the world. And the most common concentration over the counter is 200 milligram, but you can get tablets up to 800 milligram by prescription. And ibuprofen is considered a core medicine by the World Health Organization. So the WHO lists this as one of the 20 drugs you should have in your medicine cabinet. I am trying to convince you all today it should not be one of the top 20 drugs you have. And why? So we know it's very good for pain, inflammation, and fever. And the main uses are in arthritis, if people who have migraines, muscle aches. It is well known to work. And why is ibuprofen important? It's one of the top 30 prescription drugs in America, and one of the top 5 over-the-counter drugs. And the market just for ibuprofen is more than 600 million. This was last year. So it is a big market. There's lots of ibuprofen being used. Has anyone ever heard of vitamin I? So runners would probably hear this. This is a slang for ibuprofen that runners use, because when they take ibuprofen, they feel so good. They feel like they're walking on air. So it's a vitamin, but it turns out they take it before exercise, after the gym, daily. Some people take it with multivitamins. We've seen a lot of high school students taking it before they go and run long distance. So what can be wrong? The problem is that most of these people are taking NSAIDs on a consistent basis to prevent a pain they don't actually have. What can go wrong? There are 3 studies that came out in the last decade that show that ibuprofen and other NSAIDs actually do not reduce the muscle soreness following exercise. So when students take it, it's a placebo effect and they think it's working. But what does it actually do? It actually weakens the muscle. And there are studies showing there's strength loss and there's increased creatine kinase in the blood. This is a marker of muscle damage. So not only is it not helping, it's actually damaging your muscle. So athletes, please stop taking this before you exercise. And what's very clear is when you use this repeatedly, it actually can lead to severe skeletal muscle injury. What does that look like? When we take a biopsy, we see a lot of muscle scarring. So it's a problem. So how did I get into this research? This paper came out in 2011, and it was pretty interesting in that they did a meta-analysis of everything available. And they found that ibuprofen increased your chance of stroke by more than threefold. And that shocked me. I said, I'm a cardiovascular scientist, and I didn't know that ibuprofen can have this effect. So it shocked me. And then I asked people around, and no one knew this. Turns out 4 other papers came out since then, and still 90% of the population don't know this and still take ibuprofen like if it's water. So why is it causing this? I'm pretty interested. I'm also interested in this because my mother was taking celecoxib at the time, back in 2011, and developed a heart problem. I took her off of celecoxib after reading literature, and I put on something called tart cherry, and she has been on that for the last 7 or 8 years. It's something that has properties like ibuprofen, but it's natural. So first set of studies that were pretty profound was that when we take male and female mice and we give them ibuprofen for 7 days, and we give them the amount that's equivalent to taking a 400 milligram tablet each day. So this is a moderate dose. This is what most people will take, or most people will actually take higher than this. So we want a dose that's realistic. And we gave these mice this for 7 days. And then we did echo, and we looked at ejection fraction, left ventricular wall thickness, etc. Everything we can measure, and we found that there was no difference. When we separated this data and we looked at female mice, very clear. If you look at the left ventricular mass in E, no difference, very clear. But when we pass off the male, we've seen a very consistent decrease in muscle mass. We're seeing that the left ventricular mass is actually decreased. And this was very consistent. So these studies were done blinded, where the person doing it didn't know which mice had ibuprofen and which mice were controlled. So we were pretty impressed to see that this turned out this way. So we're seeing atrophy in mice given a moderate dose of ibuprofen. And this was quite shocking. We're also seeing the left ventricular anterior wall and the left ventricular posterior wall, that's LVAW and LVPW, decreasing also, only in males. So right away, there's a clear gender or sex difference between males and females with ibuprofen. So we wanted to see if we can repeat this with whole heart. And we did 2 independent studies by 2 different graduate students. And yes, we can repeat this. When we look at male, when we compare the heart to body weight or heart to tibia length, we're seeing that in males and not in females, there's atrophy in the heart. We also wanted to compare some other tissues. So we did liver, we also did kidney, and we found that no other tissue we measured would change in. So this change seems to be specifically in male heart. Now, I just showed you one gender effect. This paper came out just in 2022, and it showed that researchers at the University of Aberdeen analyzed 151,000 pregnancies over the last 30 years to see how ibuprofen affected it. And the results were startling. They found that women who use ibuprofen and paracetamol, they had an increased risk of preterm and stillbirth by 50%. Paracetamol is what we call Tylenol here. So it had a dramatic effect on stillbirth. Why I'm telling you this is because this shows you gender effects again. So there's a lot happening with ibuprofen and gender. So how do we know, what do we know about ibuprofen and how it works? So we have a well-established pathway whereby in our plasma membrane, we have lipids. And one of those lipids is arachidonic acid. That arachidonic acid is broken down by cyclooxygenases. Cyclooxygenase 1, which is called COX-1, and cyclooxygenase 2, which is called COX-2. And it breaks it down into prostaglandins. And these prostaglandins are then further broken down into other prostaglandins. And at the bottom, you can see that these prostaglandins are there that increase in pain, increase in fever. And ibuprofen and naproxen inhibits these prostaglandins. So of course, it's going to reduce the fever and it's going to reduce the pain. The problem is that if you look carefully, you'll see that there are other things there. Uterine tone, platelet aggregation. There are other things that these prostaglandins affect. So when we inhibit pain and fever, we also inhibit in other things that are important for us. So one of the first studies we did was to see, does ibuprofen cause stress in the cells? And this is ibuprofen on H9C2, which are cardiac cells. And what we found was that ibuprofen at physiologically relevant concentrations increased ROS significantly. So how do we know these physiologically relevant concentrations? When the plasma of humans who take ibuprofen is measured, we find amounts between 50 and 550 micromolar. When you take 100 milligram, you get about 50. When you take 200, it's about 120. When you take a 400 milligram dose, it's closer to 300. And when you take a 800 milligram dose, it's about 550. So the dose matters with respect to how much ibuprofen ends up in your blood and in your tissue. And you can see at these dosages, even at 100 micromolar, which is a 200 milligram dose, when we put it on the cells, it's causing like a 35 to 40% increase in the amount of reactive oxygen species that's being produced. For reference, hydrogen peroxide is a well-established reactive oxygen species inducer, and you can see that there on the left hand and the right hand side. When we look at the mitochondrial membrane potential, so in mitochondria are our powerhouse. We make our ATP there, which is our energy that we use to carry out our body functions. And we can measure something called mitochondrial membrane potential. And this tells us the potential to make ATP. And when we give even small amounts of ibuprofen, 50 micromolar, which is taking children dosage of ibuprofen, it's causing the mitochondrial membrane potential to decrease. FCCP is a known uncoupler of the mitochondrial membrane potential, and it's used as a control. What's cool about this is that the mitochondrial membrane potential, after seeing this, we went and measured ATP, and we found that ibuprofen was in fact decreasing ATP levels inside your cell, inside cardiac cells. So because we had all this data, we wanted to see what is going on in an unbiased fashion. So our lab is pretty good at something called multi-omics, where we use very high resolution mass spectrometry. And we give the mice the same ibuprofen for 7 days. Then we extract their hearts and livers, pulverize them, homogenize them. And then we use something called tandem mass tagging or TMT labeling. Why I like this a lot is because we can combine fractions from control and treated mice, or placebo-treated and other treated mice, and we can see what's happening at the same time. When you do individual mass spec, you have to do correction factors for drift and liquid chromatography changes, and we cannot get the data we get as we get here. The disadvantage of this method is you can only do it with mass specs that cost like $1.5 million or more. So it requires very expensive mass spectrometry. Fortunately, we got one of these machines in our shared facility 2 years ago, and everyone has access to it. So it's really a high-tech machine. And why this is so good? We are able to label each sample with a label, a TMT label. You see these numbers there, 126, 127N, 127C. These are all labels which are identical in mass. But when cleaved by the mass spec, it has slightly different masses, and it tells us where it came from. These are what they look like. They're very similar in structure, and they have something called a reactive group. So they'll react with the end of your peptide, the amino residues of your peptide. But they have a mass normalizer, which is what will keep them the same molecular mass. So what we add into each peptide is the same molecular mass. But under the mass spec, they separate into slight differences. So we can tell where they came from, which sample. And these mass specs are able to differentiate between 0.01 Dalton. That is the accuracy of these machines, allowing us to separate and get this high resolution. So what we do is we have 4 control hearts or 4 control livers and 4 ibuprofen treated. And then we add something that's a pool complex. And we do this because we have multiple of these that we run. So we can combine the data. The samples 5 and 10 in yellow allow us to combine data from multiple sets. The disadvantage of this is that we need computers with like 64 gigabytes of RAM or higher. But the data is fantastic. The files are like 5 gigabyte files, huge. So they're difficult to work with, but they give us data that we can't get any other way. What do? We isolate the proteins in 8 molar urea. We reduce them. We precipitate them with acetone to get rid of the urea. And then we cleave them not just with trypsin, but with lysine, because we have found that that gives us 20% more peptide. We then do the TMT labeling, and then we pull them together. So in one fraction, we can have them. Then after we have these samples combined, we can fractionate them and get really good data. We do this for males and females, and then we can combine these. So what do we get when we do this mass spec? We get over 3,000 proteins, of which about 200 are being changed in males and only about 20 in females. Huge differences between males and females. What is changing mostly in the males? A lot of cell signaling components. These, to summarize, we found that ATP production pathways were changed, mitochondrial and peroxisomal fatty acid oxidation would change, the ubiquitin proteasome system would change, and the antioxidant systems would change. This was just based on mass spec data. So we use this data then to see, can we now use this to see differences between males and females of these pathways? And we decided because we saw the atrophy. Atrophy is due to either increased proteolysis or reduced synthesis or a combination of both. And the main degradation system inside the cell is the proteasome. This is what the proteasome looks like. And those blinking lights are 6 different proteolytic enzymes in the proteasome. There are 3 different catalytic activities, each in duplicate, giving you 6. There's a beta-1, which is caspase-like activity, beta-2, which is trypsin-like activity, and beta-5, which is chymotrypsin-like activity. And these combine to cleave anything that enters there into very small peptides. Now, a protein just can't enter there because the diameter of the hole to allow proteins in is only 13 Armstrongs. So a globular protein cannot enter. A protein has to be denatured or has to be unfolded to enter. And the unfolding is done by this. So this is the ubiquitin proteasome system. It involves the E1, E2, and E3 enzyme. There's only one E1 in all of us that's active. There's about 58 E2s, and there's about 600 E3s. So the specificity is from the E3s. The E3s are what adds ubiquitin onto a protein, and that is repeated multiple times. Once a protein is polyubiquitinated, it's recognized by the 19S, the top of the 26S. It is unfolded and then sent in to be cleaved. The ubiquitin is cleaved and released. It is not degraded. Now, to convince you that proteasome is important, studies done by another group, a Japanese group, was what convinced me, and I'll try and convince you all with this. It was previously known that throughout our whole body, the proteasome is the same in terms of activity. But in the thymus, the beta-5 activity is 50% less than anywhere else in the body, just because of a few amino acid changes. They took that sequence and they knocked it in to the whole animal. That alone, so the total activity of the proteasome is still about 80 to 90%, but the beta-5 activity is 50%. That alone caused these mice, the lower one is the transgenic, these mice had arthritis by 3 months and died within 1 year. So it was clearly very important for longevity and for healthy lifestyle. And if you look at the longest lived people in the world, these are people in Okinawa, Japan. They have the most centurions. They have really high proteasome activity. So one of the questions I get asked is, how can I increase my proteasome activity? The only known things that increase proteasome activity is a compound called oliperin, which is found in olive oil, grape leaves, and olives. So when we look at proteasome activity in these mice, what do we see? We see the opposite of what we were expecting. We would expect proteolysis to increase because we see an atrophy. But what we see is in both male and female, there's less proteolysis. And this is beta-1 activity, which is the caspase-like activity, and beta-2, both in males and females. When we look at beta-5, we're seeing a trend in males, and we repeated this twice in 2 different sets of mice. We didn't see a difference there statistically, but in the female, we consistently saw a decrease in beta-5. If we look at the protein now, and this is by Western blotting, our lab has made a lot of antibodies to these, so we have antibodies that other people don't have. And you can see there that there's a clear difference between the males and the females. In the males, there's a trend towards a decrease in these immunoproteasomes, while in the females, they were all, the beta-1I and beta-5I were both upregulated. Now, I didn't mention immunoproteasome before, so you may be confused. Proteasome and immunoproteasome, what's the difference? So the immunoproteasome is what is responsible for immune function. That is what it's classified for. I'll show you just now what we found in 2014. If we look at the beta-5I, you can see there in male and in female, the beta-5I is increased in both. But when we look by Western blotting, you can see that only in the females it increased. But when we look at activity, our lab is just one of 3 labs that can measure activity of the immunoproteasome. We found that even though the amounts were different between males and females, the activity was very similar. The beta-5I was upregulated in both males and females. So why is this significant? We showed in 2014 for the first time that the immunoproteasome is what degrades oxidized proteins in our body. So when we are stressed, our body produces more immunoproteasome. So that beta-1, beta-2, and beta-5 we have are replaced by inducible subunits. They're very similar in sequence, but small changes in amino acids. And they make antigenic peptides. So they're really there to, when you have like COVID-19, to make peptides from the antigen, spike protein, to notify the body we need to get rid of these things. And we found that when we oxidized proteins and we added the immunoproteasome, it degraded very fast. This was in 2 hours, about 50% of the oxidized proteins were degraded in vitro. So when we look now, that was in heart. So when we look now at in liver, you can see similar things. The mass spec showed us that the immunoproteasome, these are really hard to detect because they're in low amounts, but with high resolution mass spec, we can see them. And you can see there on the right that the beta-5I decreased by mass spec, and we see that also by Western blotting. And you can see that when we look at females, it's a difference now. In the male, the beta-5I decreased, while in the females, it increased. We can see that also for beta-1I. So not just in the heart, but in the liver, we're seeing clear differences, gender differences. If that wasn't convincing enough, you can see here again that we see differences now in normal proteasome activity in the liver between males and females. The beta-1, which is caspase-like activity, is decreased in males, but not changed or possibly increased in females. Now, we're going to switch the talk to something that most people probably are not as aware of. So one thing that showed up is fatty acid oxidation, and I didn't know much about it. So I had to become decent at fatty acid oxidation. And to give you an example of how our mass spec works, everything in yellow is what was sequenced. The bottom is just one sequence, and everything that's red is sequenced in one direction. Everything that's blue is sequenced in another direction. So we get in two sequences to really tell us what that protein is. So we typically get about 50% or more of a protein being sequenced. So we know it's that protein, and we're really confident about it. The colored, like the green, is where they have the modifications from the links that we put from the TMT tags. Now, this is fatty acid oxidation. It involves multiple steps, and they have multiple enzymes. When we measure these, for example, FAS is fatty acid synthase, LFA BP is just liver fatty acid binding protein, so this is in liver, and CROT is carnitine O octyl transferase, which basically it transfers fatty acyl groups between coenzyme A and carnitine. FAAH is just fatty acid amide hydrolase. All of these are involved in fatty acid metabolism, and you can see there that they're all up both in males and females. These are some other things you can see there that, again, sorry, the first one was in males. They're all up in males. The top is from mass spec, the bottom is from what we measured by Western. When we look at females, we see a trend up. We see two of them are up statistically, one a trend up and the other one no change. So again, even with the fatty acid, there's differences between the males and females. So, but in males, we find that every enzyme involved in this type of fatty acid metabolism is increased. And when you see fatty acid metabolism increase, the literature told us that typically you have an increase in hydrogen peroxide as a side effect. And when you have an increase in hydrogen peroxide, there's normally catalase increase, because catalase breaks down hydrogen peroxide. So we did those experiments. This was actually done by an undergrad in the lab. She won a provost undergrad fellowship, and this was her 3 months in the lab, because she had to repeat this like 10 times, because we want to make sure it's reliable. And in the males, catalase was not increased, but it was increased in females, but hydrogen peroxide was increased in both. So the increase in fatty acid oxidation was probably responsible for increasing the hydrogen peroxide. So this is a summary of what we found with respect to the fatty acid. And you can see there where you see the up arrows, every single enzyme involved in both mitochondrial fatty acid oxidation and peroxisomal fatty acid oxidation will all increase by mass spec, and about half of them we checked by Western blotting. So this tells us a lot, because glycolysis is also affected, and we decided to look at that next. And again, we see gender changes. So metabolism is really affected. In males, pyruvate kinase is decreased and phosphofructokinase is decreased. And for anyone that remembers their biochemistry, these are two of the most important enzymes in glycolysis for rate limiting, because they're irreversible. And the most important is the phosphofructokinase. Again, if you look at the pyruvate kinase, which is the PK, LR just stands for liver form, you can see there's gender effects again. We're seeing a decrease in males, but an increase in females. When we look at phosphofructokinase, we see a decrease in males and no change in females. So huge differences. If we look at now stress-related response, so these are glutathione and glutathione transferases, things that normally are in our cell to remove stress and oxidize proteins and prevent proteins from becoming oxidized. Our mass spec told us that these were increased. We did Western blotting and we found that they were increased in males and not as much increase in females, but some were increased in females. So again, gender effects upon gender effects. If we look at how ibuprofen is degraded, so things like cytochrome, P450, etc., we can see these are actually, some of them are upregulated, some of them are downregulated in males, and in females, they're opposite.
So if we look, for example, at cytochrome 4A1, it's increase in males, but decrease in females. So again, we really have to do a lot of sex-based studies because males and females are very different. However, when you take ibuprofen, it's given the same amount, no matter what weight you are, and no matter whether you're male or female. And we envision that 10 years from now, that will probably change. This is a summary of what we found with the metabolism. And you can see there that the glycolysis, the Krebs cycle, and the fatty acids are all interfacing, and they're all being affected by ibuprofen. To finish off the talk, I just want to switch gears to oxylipids. So a lot of people don't realize that over the last 50 years, we have discovered oxylipids, and there are now over 100 of them. Why are they important? Because oxylipids are now known to be very important for cardiovascular disease, hypertension, thrombosis, hemostasis, diabetes, and the list goes on and on. Fortunately, we have one of the best oxylipid centers in the world here because Bruce Hammock has a center, and he is able to analyze 65 oxylipids out of the 100. There's no other lab in the world that can do that. The other labs do 50, and the other one did 55. So we have a really great resource. So for anyone who's interested in this, they should consult that. Why we wanted to do oxylipid analysis? We found in our mass spec that an enzyme called soluble epoxide hydroxylase was increased by ibuprofen. This is a spectra, and you can see high confidence in it. The raw data is on the right, the control. You can see the levels, and you can see the levels with ibuprofen of the intensity. And that's another way of saying the amount of that was there. So we wanted to check that. Now, there are two types of epoxide hydroxylase. There's the soluble, and then there's the mitochondrial. The mitochondrial was also increased. Now, why is this important? Soluble epoxide hydroxylase is linked to high blood pressure. It's also linked to more pain. So people who have more soluble epoxide hydroxylase has more pain, and they have higher blood pressure. Bruce Hammock's lab is in phase 2 trials with humans right now, trying to see using this as a pain reliever, sponsored by the NIH. Now, we wanted to see by Western blotting, did it actually increase? And yes, it does. So not just by mass spec, but by the Western blot, we're seeing that soluble epoxide hydroxylase is increasing. And when we look at mitochondrial epoxide hydroxylase, though, we see that it's only increasing in males and not in females. Soluble epoxide hydroxylase, which is the more important one, increases in both males and females, but only males have the mitochondrial epoxide hydroxylase increasing. When we look at activity, and this is done by Bruce Hammock's lab because it's a radioactive activity, and I don't do any radioactivity in my lab. I like my lab to be radioactive free. You can see here that ibuprofen increases soluble epoxide hydroxylase in both male and females, and it increases mitochondrial epoxide hydroxylase in both males and females. So why this is important? Epoxide hydroxylase is linked to a lot of different oxylipids. It's creating oxylipids. And these oxylipids are changing with ibuprofen. Let's take a quick look. This is just showing you some of the oxylipids on the right and the differences, whether they're up or down in males and females. But this is a better plot for y'all. You can see there the females and the males are very different. This is a PLA plot, which is basically just a plot to show you differences between males and females and between controls and ibuprofen for each group. The first one was in heart, and this is in liver. Now, initially, I told you all that when we take ibuprofen, it reduces prostaglandins that cause fever and pain. You can see here that these are the well-established ones, like prostaglandin E2. They are decreased, and that's expected. This is well-established in literature. What is not expected is other oxylipids are being affected, and in a gender-specific effect. The males have a lot of oxylipids that are increased, and the females have no change. This is a summary of this, and everything you see in green increased only in males. And you can see many of these are raw materials for the soluble epoxide hydroxylase I was just speaking about. So what is this telling us? This is another set, soluble epoxide hydroxylase that are only in substrates of soluble epoxide hydroxylase that are only increased in males. There is one change that we see in females that's not in males. You can see in blue there, the 4,5 di-HDPE, which is a product of SEH, is decreased in females, but not affected in males. So why am I telling you all of this? It's very important to understand, first of all, is there really a change between males and females? Because one of the things that bothered me was females have less oxylipids to start with than males. So these differences could be because of these just having less to start with. So we enlisted the chief biostatistician here at UC Davis, Blythe Durbin-Johnson, and she created a new program which is available online. I put it online with this publication. It allows us to compare multiple testing and be able to really tease out males and females. So we have 4 groups, males and females with and without 2 conditions, and compare them all. And what is nice about this is that she used a thing called cyclic loss normalization, which is very powerful and takes into account Benjamin Hochberg, which is really a stringent package. And from that, we're able to find that these oxylipids were significantly different between males and females. So we didn't have to rely on our eye or just, okay, this increased a lot more, this didn't increase, this did it in an unbiased manner. So huge differences between males and females. Now, why I spent some time on this? This paper came out just this year after we had published this. 500,000 people in the UK bank study, they found that people who were taking anti-inflammatory drugs, of which the major one was ibuprofen, were more likely to have pain 2 to 10 years later than people who never took drugs, the anti-inflammatory and pain-relieving drugs. In other words, when they take the ibuprofen, they feel a relief. But 2 years to 10 years later, they get back pain that was a lot worse than they ever had. While people who had the pain and bared the pain, used ice packs and other methods, they had less pain later on, 2 to 10 years later. Why we think this is happening is related to what we found. We saw that ibuprofen is inducing soluble epoxide hydroxylase. And if that is induced for a long period, it's going to increase pain because it's changing the oxylipids. So to wrap up this part, we think that these oxylipins, which are like things like EATs and different things, they are critical for the function of the cell. And in this review, we showed where it's interacting with the mitochondria and endoplasmic, the ER, and why ROS is important there. I wouldn't go into that detail. I just want to finish off with animal studies whereby we extracted cardiomyocytes from animals after giving ibuprofen, and with the help of Nepavian, Jimmy on Vat's lab in the Genome Center, using flow cytometry, we're able to see very clearly that cardiomyocytes from ibuprofen-treated mice have huge increases in ROS. Not just that, the fibroblasts in those hearts also have huge increases in ROS. If we look at the proliferative capacity of fibroblasts, we see there's a trend towards an increase, but it wasn't statistically significant. So the fibroblasts seem to be able to proliferate. They just are producing a lot of ROS, just like cardiomyocytes. So that's telling us what is happening. Where is this ROS coming from? We think it's the mitochondria. Why? We isolate mitochondria and we measure complexes. Because I'm a biochemist, I'm not a big fan of the seahorse because it takes 30 minutes to get to some of the complexes. I like to isolate the mitochondria and do each complex individually. And we find gender effects. In complex 1, we find that for complex 1, from female hearts, it is inhibited by ibuprofen, while it's not inhibited in male hearts. Now, complex 1 and complex 3 are the main producers of reactive oxygen species in the mitochondria. They produce superoxide ions. Making of ATP is not efficient, and there's about 2 to 4% inefficiency produced in ROS. If we look, these are in cells now, so both in cells and in the mitochondria isolated from animals, we're able to reproduce these things. In cells, we see that even complex 2 is being affected by ibuprofen. So it's affecting different complexes. And this is a visual representation of how ibuprofen can induce ROS. And you can see there by giving ibuprofen at 200 micromolar, which is someone taking like a 200 milligram dose, a fairly weak dose. You can see there how it compares to a control. And in this case, the control was hydrogen peroxide. And you can see very clearly that it's significantly above the background. Now, the good news is, in a test tube, we're able to prevent all of these. We can see cell death, cell viability, this study here. We can see ROS. By preventing that ROS from the mitochondria, we see the cell surviving. So whether we give something like mitotempo or ascorbic acid, we can see the cell being rescued. So we do think that all of the side effects of ibuprofen we are seeing, or most of them, is due to the production of superoxide ions. When we give vitamin C to animals, it didn't work. It turned out that mice produce their own vitamin C. They're very different from us. And this data is buried in the literature. However, our recent studies, when we give vitamin E, we're seeing a lot more benefits now. We're seeing a lot more preventative. We see a lot of the side effects of ibuprofen not showing up anymore. So the good news is antioxidants, other than vitamin C in mice, but probably in us, since we don't produce vitamin C, may be beneficial. So I would like to finish off just by letting you know that we think that ROS is what's damaging the proteasome. We have data for that. It's what's producing oxidative stress, and it's what is causing the cell death and eventually causing the cardiovascular disease. With that, I'll just quickly summarize 2 things that I think is important. Long-term low doses of ibuprofen, even small amounts, taking it for long-term, impair the proteasome. We have that from cell data. And other tissues besides the heart and liver are affected. The kidney is affected. Skeletal muscle is affected. So ibuprofen is not just damaging your heart, it's damaging many tissues. And with that, I would like to thank the members that were involved in this, Shushita, Gabriella, and Rashid, as well as our collaborators, Bruce, Christoph, Jun Yang, and Blythe Derwin Johnson, and Nipavian Shimon Vat should have been on here, and funding from NIH. And with that, I wanted to leave some time for some questions. Thank you, everyone.
Thank you, Andre. Thank you so much. Very nice talk. So let's go for questions. Do we have questions there? Do I see a hand? I think that Crystal has a question. Go ahead.
I do. That was a really interesting talk, Aldrin. Thank you for that. I have to say it's a real big bummer because I love ibuprofen. So I don't know if anybody else feels like that it works so well. But the question I had, and I was actually asking my students, we study cyclooxygenase pathway, and we find that, there are all sorts of detrimental effects when we block it molecularly that phenocopy the ibuprofen exposure. And I'm wondering if you think that maybe inhibition of cyclooxygenase itself, not just by ibuprofen, but if that pathway inhibition, it might also increase reactive oxygen and have the same sort of phenotypes.
Very good question. And we have done that experiment. It does. When we use cyclooxygenase inhibitors that are not related to ibuprofen or other anyone we use, it produces ROS, right? What we're trying to figure out is, and the ROS tends to be from the mitochondria mostly. We're trying to figure out, is it getting into the mitochondria as ibuprofen or something, or whatever we put in, or is it a metabolite? Right now, it looks like a metabolite, and it looks, data with acetaminophen using our mass spec suggests that acetaminophen is easier to measure because we can put a tag on it. We're still fighting to put a tag on ibuprofen, but with acetaminophen, it's actually binding to mitochondrial complexes. That's why it's causing the activity decreases. But with your question, the COX is something different, but that by itself causes ROS. All right. The problem there is that I'm sure you're familiar with the field saying there's a COX tree also, right? So that complicates things because there's a quasi form that seems to show up sometimes in some labs and in other labs it doesn't and stuff. But for others who are not really into COX, this is not something important. But for us, it seems pretty complicated. But a bigger question along that line is that full inhibition of COX is very dangerous and it will cause death. So that's why I actually recommend to people taking children's dose of ibuprofen. 100 milligram of ibuprofen is what I use for a headache, and it works great. And I had my lab test it out, and they said, yeah, it's working. And it turns out studies done in the hospital found that doctors were prescribing 800 milligrams too often. When they did a study, they found 400 milligrams was working as well as 800. 800 added no benefit, but it added so much this side effects that people don't take into account. So amount and duration matters.
Okay, thank you. Any more questions? Thomas, has you?
Yeah. So thank you, Aldrin. This is like this is a familiar topic. I'm an ultra runner, and so this idea of the danger of ibuprofens is something that's been circulating in the sport for a couple of years. In fact, it was in 2021, a rather large race organization in Europe, UTMB, banned all non-steroidal anti-inflammatory drugs. So I have two questions, So one, you persistently referred to ibuprofen, and I'm wondering if you believe that this, the dangers of ibuprofen is the same for all non-steroidal anti-inflammatory drugs. Like the other one, like Aleve has a partnership with the Ironman World Championship organization that a lot of people are questioning the appropriateness of. And then two, like it's, as I understand it, it was banned from UTMB, not because it's a performance enhancing, although perhaps there is some of that if it kind of masks pain that allows you to kind of push through, an activity in an endurance event. But it's not necessarily like on the same level as like a steroid, perhaps. So it's performance enhancing. But are you aware of there being any efforts like within organized sports to ban this more broadly, not necessarily because it's a performance enhancing, but because of the potential dangers to athletes that they're not, aware of, or that they have a kind of a regular habit of kind of preloading on this sort of stuff in order to prevent pain?
So very good questions. Your second question, so ultramarathons is the one that took the first step because apparently in the literature, it was being abused the most by ultramarathoners. Everyone pretty much was taking it. And there's actually a fantastic blog that I read like a year ago by an ultramarathoner whose title is, "Please don't be like me." And then he went through, when he was taking ibuprofen, how great it was, and then how bad it was when he realized what he did to himself and what he does now. And he is a firm believer in, so he blends berries and stuff and eat these smoothies with, where it has to have the fiber and everything still in it. But he uses that before his ultramarathons now, and he claims, there's no scientific data, but he claims that it makes him so good, and he can replicate everything. But I want to just say something very clear. The data recently showed that it's a placebo effect. There's no enhancing function of ibuprofen, none at all. The data was pretty clean to me that this is in the mind. If we give people a sugar pill and tell them that's going to help them, 50% of people are going to do better. Because hey, a doctor told me this must be good. Or other people are doing it, so I must do it. In fact, and there was actually more damage. So when they biopsied, there's a paper showing biopsies of several ultramarathoners who took ibuprofen, and they had so much damage to their muscle. And there was one paper that showed that ultramarathoners who took ibuprofen had 50% increase in rapid onset kidney disease. It appeared in hours after the ultramarathon, and that was pretty frightening. I'm sure you probably heard of one or two people who had that.
Yeah, it was the kidney. I hadn't heard before the cardiovascular damage or and just the more general tissue. Is that they were really hammering the kidney in part, I think, because it related a lot to the change in the blood sodium level and hydration, dehydration that is so prevalent in our sport.
Yes.
Thank you.
So my data is suggesting it's not just damaging the kidney or the liver, it's damaging everything. The only benefit we are seeing is that it kills cancer cells too at a slightly faster rate. So it is beneficial in preventing cancer, but the benefits don't outweigh the risks of taking the high doses. But your first question was, do other NSAIDs behave the same way? No. Naproxen is much safer, probably like 4-fold safer. So in our hands, so we actually did naproxen and we did meclofenate sodium, we did diclofenac, and diclofenac is a lot worse than ibuprofen. In fact, diclofenac is responsible for 90% of all the vultures in India dying. It is that bad. They used to give it to their cows because they're sacred, and when the cows die, the vultures will eat them, and they will concentrate the diclofenac, and they will get liver dysfunction and die. Now, when you look at a vulture, they look stronger than us, right? So it shows you, so diclofenac was the worst, then ibuprofen, then naproxen, and aspirin was very safe. It didn't produce ROS at all. But of course, it doesn't have that much pain relieving effect. So each NSAID is very different.
Okay, Lina, do you have a question?
Yes, I do have a question. Well, first of all, thank you very much for your presentation. And earlier you had mentioned how, like if you take it long-term, what that does to a body. Can you specify for me what long-term means?
Okay, so clinically, yes. Clinically, long-term is 12 days or longer in a row.
12 days.
Yeah, so
Okay.
the current literature suggests if you take ibuprofen every day for 12 days, you have the increased risk of stroke by 3-fold for up to a year after, even after stopping it. If you take it one day, the risk is like for 24 hours and it goes away. But as you start going long-term, the risk last is also long-term. So if you're taking ibuprofen for like a month straight, you probably have a risk for 3 years, even after stopping it. So some doctors recommend, who I've been talking to, they give their patients acetaminophen one day, ibuprofen the next day, then naproxen the next day. They swap out because they all have different features.
Oh, thank you. And my second question was, in there you also mentioned antidepressants as the same. Can you say a little bit more about that?
Yes. So what is not known is that, what a lot of people don't know is that you cannot take ibuprofen with a lot of other drugs. You cannot take ibuprofen with ACE inhibitors. You cannot take ibuprofen with aspirin. They all have consequences. You can't take them with the antidepressants. And if you go and look at any water in California or in America, 2 of the top compounds we find is antidepressants and ibuprofen in the water, right? And people are mixing them. And if you just do a quick search, you will see that combining them is dangerous for our health.
Are doctors aware of this? Because
I'm just thinking in my limited experience of having been prescribed ibuprofen, like after giving birth, for example, I mean, I was on like 800, whatever the units are for ibuprofen for an extended period of time. And they just said, just keep eating them, you know? But I had never heard the antidepressants and ibuprofen. Is there an effort to educate the medical field, like people who have contact with patients, to notify them of this or put in some kind of like warning within their systems when they're prescribing something to let patients know?
So doctors who are doing this are seasoned, and they think that it has been used so long, it's safe. Even though the number one cause of death in people over 65 is heart problems, and they're the ones who take the most ibuprofen and NSAIDs. They don't see the link. So what I am doing is half of my lab undergrads go on to medical school. So I had them write, we're writing different parts now that's going to be in a chapter of a book, and they're all involved. So it's about 40 students over 3 years. So that they, when they become doctors, they can think for themselves and realize to start the cycle of stop overprescribing, because it's not just, so it's okay to prescribe ibuprofen, but the amount 800 is not needed. 400 works. In the studies that were done, 400 works well. And for people who take 400, 200 seems to work for a lot of them. So the doctors don't titrate people, and they should. They should be telling people, start with 100 milligrams. If it doesn't work, use 200. The lowest dose and for the shortest time. And there are other studies showing that when you give something like not normal Bing cherries, they reduce the amount of ibuprofen you need because they have natural pain relieving effects.
Wow. And another question I had was, did you notice that the overuse of ibuprofen was found to be like more noticeable in the US than in other countries? Is it a cultural thing?
The data for that is not clear, but what we do know is that 15% of people who take ibuprofen take more than the recommended dosage, which is a big problem. Those are the people who are going to have the heart problems showing up very quickly, liver problems, etc. And you may not think 15% is a lot, but 80 million people in America take ibuprofen at one time or the other. And 30 million people take prescription ibuprofen, which is 16 million people. 30 million prescriptions are written for 16 million people, which is a lot.
Wow, so interesting. So any more questions? Do we have more questions today?
I just wanted to say, I mean, this is particularly bad news for women who use ibuprofen for menstrual cramps, right? So that's at least once a month that systematically that women who have debilitating pain from menstrual cramps are taking something for their pain, and it tends to be ibuprofen. And it's just normalized that that's what you do, that it's fine. And at the same time, I mean, of course, maybe certain athletes, I think in particular, ultra marathoners or marathoners, I mean, they have a high threshold for pain because running those long distances produces a lot of pain in my experience, and they can do it, right? But for people who suffer from severe chronic pain, there's a lot of those. What are the options?
So we have done extensive literature research on what's possible. And when it comes to fever reduction, there's no alternative. But when it comes to pain, there's actually a lot there that's natural products that will help you reduce your dependency on them. And you can do a quick search on black cherry or tart cherry. They were initially found to be very good for gout. But a study was done where they look at the antioxidant capacity and the anti-inflammatory capacity, and it was near ibuprofen levels. And I have tried it myself, and when I take it, anytime I'm feeling sick, I take it as a tablet form. You can get it in Trader Joe's too as a liquid form, and it works. It reduces your amount of any other thing you need to take. So we can't completely get rid of it, and that's not what I'm trying to advocate. I'm just advocating taking the least amount for a shorter time as you need it. So during during a period, it's okay to take it, but try to take it only for that day you have it the worst time, while you take other things when it's not as bad. Or if you have it for 2 or 3 days, take it only for those days. Don't take it like some people take it for 5 days, you know, straight. Try to minimize. Just be cognizant of the fact that it has consequences the longer you take it and the higher the dose. So don't live a miserable life. Don't just wallow in pain. You know, take it when you need it, but just be aware that abusing it, like many students do, is not something you should be doing. And there are other things. There's actually about 8 compounds that are natural that reduce pain naturally to about 50% of what ibuprofen does.
So any more questions? Looks like we are done for today. Thank you so much, Aldrin. This was a fantastic talk and a fantastic discussion. So thank you so much, and see you all of you in 2 weeks. Okay.